This is a tremendous honor. Good morning. So today, as digital and physical worlds collide, at the boundary, there is a veritable tsunami of data. Literally, we are swimming in sensors and drowning in data. When confronted with this challenge, today's computers are really literally overgrown, highly power expensive calculators. In contrast, the human brain has awesome powers of sensation, perception, action, cognition, and interaction. When confronted with an object such as a rose, it can extract from the object its appearance, its smell, its texture, its taste, and various sounds. It takes this multidimensional sensory data stream and converts it via a series of spikes or action potentials or firing on and off of neurons into streams that the brain merges. And brain then can act via various motor modalities such as speaking, hand, feet, as well as a couple non-polite motor modalities. And it can act in an ambiguous environment in a context-dependent fashion. And it can do all this while consuming less power than a 20-watt light bulb and occupying less volume than a 2-liter bottle of soda. So this really is the dream. Can we mine Mother Nature's patent fund to create a new generation of brain-inspired computers that can rival the function, the power, and the size of the brain? To this endeavor, we have built a team and a research effort spanning disparate, very far-flung disciplines of neuroscience, supercomputing, and nanotechnology to create an entirely new field called cognitive computing. And it is my intent today to tell you one new result in each of these three areas. Before going forward, I would like to acknowledge that this research is being sponsored by the visionary initiative called DARPA Synapse, which was the brainchild of Dr. Todd Hilton. Also, our project brings together IBM, universities, and government. From universities, in our phase 0 and phase 1, we had Stanford. We're continuing to have Wisconsin-Madison, Cornell, Columbia, UC Merced. At IBM Almaden, IBM Yorktown Heights, Zurich has been involved, India Research Lab, IBM's fabs in Burlington, East Fishkill, as well as our lab in Austin. We also had support from Department of Energy and has collaborations with Lawrence Berkeley National Lab, Horst Simon, as well as Lawrence Livermore National Lab, and used a supercomputer at KAUST. And I would also like to thank a large number of our phase 1 team. And before I go any forward, we are hiring, so if you want your name to be added, I would very much appreciate qualified people involved in asynchronous circuit design and as well as other aspects of neuroscience, supercomputing, and nanotechnology. So let's pick up our story. Marian Diamond and Arnold Schiebel said that the human brain is the most complex mass of protoplasm on Earth, perhaps even in our galaxy. Now, what underlies the complexity of the brain? At a very simple mathematical level, one can think of the human brain as a graph, as a network. The vertices of the graph are the neurons. The edges of the graph are the synapses, which are the junctions between the neurons. Given that everything that we are and are able to do and feel comes from this network can be studied via two disciplines. First is the structure of the neuron, which is known as the neuroanatomy, and second is the dynamics exhibited by the neuron, which is studied via neurophysiology. So it's really the combination of structure and function that gives rise to behavior. So let's first focus on the structural aspect of the brain network. This network can be studied at a variety of scales. It can be studied at the level of individual synapses. It can be studied at the level of gray matter, cortical circuitry. So if you take the cerebral cortex, it opens up and unfolds, has about 2,400 square centimeter in surface area, and about 2 to 3 millimeter of thickness. So it's a 2 plus epsilon dimensional technology. If you drill a hole through the cerebral cortex, what you find is the neurons are roughly organized in a stereotypical canonical organization, which consists of several layers or laminars or stratas. And this organization seems to be consistent across different parts of the brain, dealing with functionally segregated, functionally different tasks. It also seems to be more or less invariant, or at least let's say consistent across the mammalian evolution scale. So you can study it at that level, or you can study it at the level of long distance connections between the brain called the white matter. The reason is because the long distance wires, the axons that carry the neuronal firing, are coated with a cell called myelin, which is white in color. Okay? So in particular, what you see here is sort of the very state of the art, it's about 100 years old, of how white matter in the brain is studied. And Nicholas Steno in 1669 referred to white matter as nature's finest masterpiece. Now what you see here is a joint work with Professor Brian Wondell at Stanford University. So the idea is you take a human brain and you want to study its connectivity. You put the brain into a large magnet. The magnet is rotated, which causes the water to diffuse, and the water prefers to diffuse through existing pathways than across cell bodies. This diffusion selectivity can be measured at the level of small voxels, about 1 millimeter by 1 millimeter by 1 millimeter. Could be smaller, could be slightly bigger. So the entire brain is cut up into such voxels, and you have a diffusion tensor representing the selectivity in each of the voxels. Then you use statistical algorithms, for example, spline fitting, so forth, to try to conjecture a pathway that puts together the different diffusion selectivities to make a hypothesis as to wiring diagram of the brain. Now, diffusion tensor imaging is very important because it's the only non-invasive technique for understanding the wiring of the brain. However, today, it suffers from lots of false positives and false negatives. So anatomically, the gold standard for understanding the long distance connectivity in the brain is via tracing. And there is a database called COCOMAC, which compiles about 400 research studies spanning the actual anatomical tracing in the macaque monkey brain over last 50 years. So this is hundreds of experiments, hundreds of animals, probably many, many hundreds of neuroanatomists working together to meticulously, painfully, and conscientiously mapping one wire at a time. So this was the COCOMAC database. The database was full of inconsistencies, conflicts, so forth. So after about 4 years of work, trying to put this entire database into one common framework, I'll tell you a series of results. What you see here is the hierarchical parcellation of the brain into different lobes. The occipital lobe, which is the visual cortex, parietal lobe, temporal lobe, frontal lobe, insula, cingulate cortex, thalamus. Now, thalamus is the relay station that connects sensory information from the senses to the cortex and relays motor information back from the cortex back to the motor output, as well as basal ganglia, which is the seat of 4 F's: food, fight, flee, and mate. So what you see here is occipital lobe. The occipital lobe has been divided into a series of hierarchically smaller regions. And you see V1 at the upper right corner there, which is the primary visual cortex. That's the region where light from the retina first gets to the cortex. Similarly, we have temporal lobe that we were able to extract, insula and cingulate cortex, parietal lobe, frontal lobe, thalamus, basal ganglia. And now as you take these different pieces and put them together, a fuller picture of how the brain is decomposed into different regions begins to rise. Okay? So what you see here is a hierarchical decomposition of 383 brain regions in the macaque monkey brain. Each colored rectangle corresponds to a little physical patch in the brain. Now, this just gives you the vertices. What about the edges? So what you see here, if you strain your eye, you just see a single wire on the right side of the screen connecting visual cortex V1, primary visual cortex V1, to thalamus. But our network has not one wire, but 6,602 edges. Okay? As you can see, it's a jumble. It's very difficult to see it. So by using a very beautiful technique from Denny Halton, wherein you can bundle those edges with common source or common origin, we're able to refine the graph until a fuller picture emerges. So the white connections are the white matter. Okay? So given this collection and the network, it's interesting to compare it to notable past collections. So the most famous and most analyzed network is from Feldman and Van Essen, circa 1991, consists of 32 areas and 305 connections spanning the visual cortex. You can see that our network, compiled from COCOMAC, has 10 times more brain regions and 20 times more connections. So we are finally getting a better view into the complexity of the most complex object in the galaxy, according to Marion Diamond and Siebel.